Actually, I'll just to kick off. So welcome everyone. To our last seminar of this term, but I wanted to also just flag up, we've got lots of really exciting speakers lined up for next term already. And a whole range of topics from land use justice in the UK through to the future of the Congo Basin and many other themes in between. So do sign up for that if you're not. And it's particularly grateful for our speaker today to step in because the planned speaker, Robert Fish, had to cancel because of some planned medical appointments that came up. And so he'll hopefully appear next term. And so it's great to invite Francesca Vitali, who's from Smithsonian's National Zoo and Conservation Biology Institute and has lots of interesting things to tell us about the link between animal health and animal translocation in the context of Africa. So welcome.
Thank you so much. Can you hear me? Yeah. Thank you so much. It's a pleasure to be here, and thank you so much to Jesus and Javinder for inviting. It's a big pleasure to talk about rhinos and the work we are doing at the interlinkages between wildlife health and conservation translocations. Can I use this to change slide? Okay. So before we enter the core of the topic, I would like to tell you a little bit about myself and why I really care about this topic. So I'm a veterinarian, and it's a bit unusual for veterinarians to go into conservation medicine, into conservation, especially when I was a student. We didn't know much about conservation, to be honest. We were studying domestic animals, and I wanted to be a horse veterinarian. But I went to Kenya for a summer internship, and I ended up at the wildlife orphanage of Nairobi. So I was feeding the animals, and this is orphan lion, very sad story. Her name was Sarabi. And her mom was killed probably, and they found Sarabi on the way to Somalia for the illegal pet trade. And when I was at the orphanage volunteering, I met this guy, Dr. Edward Kariuki, who is a veterinarian. And he told me he had to do health assessments on the lions at the orphanage. They had many orphan lions there. They had about 25, all from the illegal pet trade, at least most of them. And at that time, I was also very passionate about anesthesia. So talking with my university mentor, the one up there, Professor Giuliano Ravasio, who was also passionate about lions, but zoo lions that were coming to the university for CT scans and so on. He decided to support me to go back to Kenya and do a study on lion anesthesia. And when I was supporting Dr. Kariuki to do his health check assessments, also some wild lion anesthesia came up. And from a veterinary point of view, these two guys are very similar. What we tried is protocols in lions that we usually use in cats because the anatomy and the physiology is extremely similar. But I learned very quickly, let's see if this goes. Anyways, no, it was a funny video. It was just to show animals waking up from anesthesia, like in this case, a rhino. So I learned very quickly that a good wildlife anesthesia is essential to being a veterinarian in the wildlife medicine space. Well, you can see this now. And this happens a lot, actually. It's not rare. So I decided to do a PhD on this topic, again, with the University of Milan. And during my PhD, I studied different species, for example, giraffes. This was also my first National Geographic grant. And giraffe anesthesia is very complicated. They have a unique anatomy and physiology. We don't know much about it. And also you see people seated on the neck because they're so sensitive to anesthesia that actually in the field, we reverse them immediately. So they're technically awake. So this giraffe is awake and there are people sitting on the neck so it doesn't stand up. But this is, for me, is not a good anesthesia. So I was researching how can we make it better also for the welfare of the animal. And then I also studied buffaloes along the same line. on the left, you have a buffalo that is partially awake. You can see all the ropes. Rangers are struggling to keep the buffalo restrained. And on the right, you see with the new protocol, we studied the buffalo is sleeping and the rangers are chatting because they don't know what to do. They're like, okay, this is so easy. So technically, wildlife anesthesia is called chemical immobilization because we immobilize them chemically. And I would like to take you through very quickly on why it's so complicated and why it needs studies and why it has high morbidity and mortality. So first of all, you have to think about the fear. The animals don't know that we are immobilizing them. And some animals are extremely shy or aggressive, like the black rhino. So they have a fight or flight response. And then overexertion. So when you go for a run, your brain will tell you at some point to stop, right? You feel tired and you will stop. But these animals have so much adrenaline that they will not stop. So they go into oxygen debt and muscle fatigue. And this creates, for example, some electrolytes to go into the bloodstream, like potassium, and that can kill the animal instantly. So this is how bad it gets. And then we give them the most potent opioids that are available because these are huge animals. And in order to make sure they're sleeping, these are the drugs we have also for concentration to put it in a dart, in a 3 ml dart. And these opioids are amazing in terms of restraining the animal, and they have a lot of properties, but they also have horrible side effects. Among these, there is hypoventilation, so the animals are not ventilating because the opioids tell the brain, stop ventilating. And also within the lungs, like oxygen get into your lungs and then exchange with the blood vessels, but the opioids make this exchange change. So the gas exchanges are not happening the right way. And this is what happens. So they already did a marathon. They need oxygen, but then we have the opioids that tell them don't breathe the oxygen. So disaster. And then recumbency in big animals, the gravity creates a further change within their lungs and their vessels. So this further improves worsen things. And then most of these animals have a unique anatomy and physiology that we still don't know much. So what do? We extrapolate knowledge from other species, and there's nothing worse to do because every species work in a different way. So we can make it even worse. Result, we can have a hyperactive mortality, delayed capture morbidity, which very often we don't know because very often we mobilize these animals, they're wild, and we don't know what's the effect. And we also get chronic stress. Again, we don't know to what extent, what is the result of all of this. So this is another study I did during my PhD, and since today we will be talking about rhinos, I just wanted to make you understand a little bit, for example, the goals and the objects. So for example, with the black rhinos, something that happens very often is that we extrapolate from the white rhino that is much more studied. So we don't know as much as in white rhinos for the black rhinos. So we decided to study if the drugs we give to white rhinos, some drugs are given when the animals are already sleeping, if they improve things also for the black rhinos. And also, for example, this monitoring system you see here is taken from human medicine. And I started using it, but now the question is like, is it accurate? Can we trust it? So we were also studying this monitoring. And again, we're not going to go through this, but just to make you show you very quickly some results. We saw severe metabolic acidosis, which is something that can be extremely impactful on the health of the animals. Our animals were hypoxemic, so it means not enough oxygen in the blood. They had increase in stress markers, and this cardiac troponin you see here that is usually used in human medicine as cardiac biomarker sometimes for heart attacks was increased. But then we don't know the value of this because we don't have enough research. Sorry. So when I was doing my PhD, a lot of the animals that were immobilized were also actually translocated. So my question was, if the animals, when they're sleeping, they're so compromised, what happens when we put them in a transport crate for hours? after. You know, they're already compromised, and it's not that we are going to release them and they're going to go back to their herd or their prides, but they're going to go into a transport crate for hours. And for example, these guys, they stayed in a bomba for 3 weeks. We were feeding them for 3 weeks. This is all stress. So I started looking in literature, and there's not much. So I started to be seriously interested in this topic. I want to know more. As a wildlife veterinarian, I'm responsible for the health and welfare of these animals, and we don't know what we're doing to them. So in the past 6 years, we've been building this project on conservation translocations and wildlife health. So everything I will talk about now is the ideas of myself and several other people. We work together with the Smithsonian, with the IUCN Conservation Translocation Specialist Group, the government, Kenya Wildlife Service, and Wildlife Research and Training Institute, my other affiliations, which are Impala Research Center and International Livestock Research Institute. And then we work with VetMed Uni and the University of Pretoria. And these are all the sponsors that up to now have been supporting our project. So now I'll start a little bit talking about conservation translocations. I don't know if you're familiar with the term, but conservation translocation is the movement and release of an organism with the primary objectives of being conservation. So commercial translocations that sometimes we see, for example, from Southern Africa are not conservation translocations. So the goal is different. So according to the IUCN, so these are the guidelines from the IUCN Conservation Translocation Specialist Group, there are 4 types, which are population reinforcement. For example, we do this a lot with the rhinos. We bring in new individuals in sanctuaries to expand the genetic diversity. We have reintroduction, so species went extinct in a site and we bring them back, which also happens a lot with the rhinos. We have assisted colonization, so we're moving species to a new area to help them adapt. For example, in case of climate change, this is happening more and more. And ecological replacement. So species went extinct, and we bring back a species with a similar ecological function for ecology purposes. And all of conservation translocations have increased in the past 30 years of 30 folds, according to the IUCN, across all taxa. So they're becoming extremely common. And also they're becoming relevant at global level. So conservation translocations are mentioned in the Global Biodiversity Framework, in particular in Target 4, which is about halting species extinction and solving human-wildlife conflict as well. And they're cited as the only tool for some species to get recovery. So they're really getting a lot of attention. They're becoming seriously recognized. And also for Target 2, that is about restoring ecosystems, because for doing real restoration, we need the flora, but we also need the fauna. However, there are no monitoring indicators. And if ideally conservation translocations are the tool to solve conservation and sometimes restoration, we are not there yet. We have many flaws. So we have to think how to make things better. And from a health point of view, translocations are extremely complicated. So we have different phases. We have capture, captivity, transport, and release to a new environment. And across all these phases, through different physiological pathways, chronic stress can build up. And for example, as a result, we can have animals that are starving, they're more susceptible to disease, or they're spreading disease much more. Their reproductive capacity goes down before of, again, chronic stress. They're more susceptible to predation, or they're not dispersing the right way. And all of this can build to translocation failure. We have some knowledge. This is a review from my colleague, Frédéric Paulin, who is a member of this project. We know, for example, that capture and transport are playing a main role in the stress mechanism, that stress affects animal health, but also behavioral competence and cognitive processes. Stress can also have a cumulative effect. For example, in Kenya some years ago, they did a study and they saw that translocated black rhino females have decreased breeding performance. But we really have major data gaps. across species on survival rates, demography, behavior, disease incidence, reproduction, space use, and settlement. So we need to do better. So I would like to now ask you to think like an animal. I don't know if you can see very well here. It's a lion being released. And if you were this lion being released, what would you do next? Like, where would you go? What would you do? You know, you've been like on a transport for like 12 hours. You've been captured. You're confused. Like you heard, you know, new smells, new noises. You're with other lions, and then you get released. And again, think about what's coming before. Helicopter chasing you, it's like an alien machine. Basically, they don't know what is it. They don't know they're going to a better place. And then anesthesia, sometimes they wake up. Transport crate, release. And then we move this animal, asking them basically to reproduce, you know, to expand their population. And we're not really understanding the whole picture. So again, what's the survival of the animals? What's the reproduction? What's the effect on population? But also on the other side, what's the effect on the other animals? Like if you bring back lions in a place where they have not been, or rhinos, of course there are, you know, more things happening. Or what's the effect on the livestock, on the humans, and on the ecosystem? And these are all questions that we don't have an answer. And again, we have to remember that we're doing this because we are claiming there is a biodiversity loss ongoing and environmental changes. So we want to help species and the planet, but we don't know what's the morbidity, the intergenerational impact on moved animals, and the mortality. And as a veterinarian, for me, it's an ethical problem because we put animals through a lot of stress, but it's also a conservation problem because sometimes these animals don't really contribute to conservation, restoration problem, looking bigger, and also a public health issue. And I don't know if you're familiar with the concept of One Health. It's been discussed more and more, and it means that the health of the ecosystem, humans, livestock, and wildlife, they're all connected. And if we think like this is Nairobi, and we have the park and the city bordering. And this is usually wildlife areas in Kenya. You have the wildlife and you have the pastoralist communities that live with the wildlife. So if we bring animals and their diseases, and these animals are stressed and they're just spreading disease even more than usual, this disease can go to their cattle very easily. And if their cattle starts to die, it really affects the communities. So we need to have a big picture in mind when we do conservation translocation and think about all the consequences. And this is a CITES database on legal trade transaction in cited listed species. And first of all, you can see throughout the years how much it's increasing in terms of like movement of animals. But also this is CITES, so this is only for some species and is across borders. We don't have any database either like intra-borders that show how many translocations are happening or where the animals are going. And I think this is extremely worrying to not know all these movements happening. So let's go back to the rhinos. I'd like to explain to you why I chose the rhino as a species. The Eastern Black Rhino is a subspecies of black rhino, and it's critically endangered. There are slightly more than 1,000 individuals in the world, and most of them are in Kenya, like 95% of the population is in Kenya. And in Kenya is a very important species. There is a recovery and action plan for the black rhino. And currently rhinos are in 18 rhino sanctuaries. There were 16 up to 3 years ago, and now they're really trying to expand the population. Indeed, they have a new target of doubling the population in about the next 10 years. So from 1,000 to 2,000. And to do this, also the veterinary services are involved through identification of all the animals and translocations. This is how a rhino sanctuary looks from the sky, as you can see. So the road is where there is the fence. And as you can see, looks like the same inside and outside. And there is a reason, because all the other species can cross There are either corridors or can go through this the rhino fence. They can go through the rhino fence, but rhinos cannot pass through. So this keep them safe because there's been like a very bad poaching crisis throughout years. So Kenya is doing very well through this system, but they need to keeping them safe. So these are the identification systems through ear notches. So depending on where the notch is a different number. And all the rhinos get captured around 2 years old, 2-3 years old to get identified. And they also put a radio transmitter here in the horn. So they put glue on it, and rangers go out with an antenna to look for the rhinos. Black rhinos are very shy, so they hide very well. So that's why they have antennas. And recently, they also started to put GPS tags. So for the rhinos, there is a system in place in which we have rhino rangers in the rhino sanctuaries in different sections, and they go out every day looking for the rhinos. And if a rhino is not seen for a few days, so you can see here's the headquarters with the position of all the rhinos. And if the rhino is not seen, they send the plane to look for the rhino. And this, again, is measures against poaching. And this is how translocation happens. So we use these rhino crates. The rhinos are basically they are anesthetized, and then they're given antagonists very close to the entrance of the crate with ropes. And when they start to waking up from the anesthesia, they're blindfolded. Everyone push them in, and there is a rope from the front taking them in, and then we close them in these crates. And then for release, either we do self-release in bomas, where they usually stay around 1 month, or hard release, so directly in the new environment. So as you have seen, there is a lot of resources allocated to rhinos and also a lot of monitoring through the rangers. So we have decided to start a project on understanding the outcomes of translocation in rhinos. And we have a project through the Smithsonian Institution, the government of Kenya, Kenya Wildlife Service, and the Wildlife Research and Training Institute, and the Impala Research Center, which is this research center in the middle of the bush where Smithsonian has a lab. And we have now an MOU that will last 10 years, and the project is growing. So it's more investigators on the same project. And our research goals are to establish safer veterinary protocols, starting with the anesthesia and the translocation management, understanding the acute and long-term health consequences of translocations, to detect risk factors for morbidity and to understand how to prevent these risk factors, and to standardize indicators in order to facilitate, even when the project will be over, to facilitate monitoring for the government, so they have like easier indicators to follow the rhinos. So here's where we start. If possible, we start before the capture. Sometimes we know which candidates will be moved. Sometimes we don't, because of several reasons. They change, sometimes the same day of the capture. So we collect all the information from the capture, including the behavior of the animal, of course, drugs we give, the length of the chasing, and so on. And then throughout the anesthesia. Anesthesia is usually very quick, it's 15 minutes. So this is what I use for anesthesia. This is to measure the oxygen levels in the blood. It's a pulse oximeter, and as you will see, it's different from the human one because rhinos have a thick skin, so it has to go inside the eye to read. And this is the capnometer. It measures carbon dioxide. This is my co-PI, Matthew Mutinda, who is also doing his PhD on anesthesia, and we work a lot together on the anesthesia part. And then during the capture, we take sterile samples from arteries. So we need arterial blood to understand the gases in the animals and to really understand how the animals are doing from several points of views. And then we also take venous blood to analyze various things from biochemistry, stress biomarkers, measures of dehydration, of muscle fatigue, and so on. And we also collect saliva samples to understand the acute stress. And the reason is that in saliva, cortisol is released very quickly in about 20 minutes. So we can have basically baseline and the change with the capture. And sometimes we analyze samples directly in the field, like here is my biochemistry machine attached to the generator. And sometimes we go in the lab, depending on what we analyze. And then we established a system to get fecal samples for 6 months after the animals are moved. We also have to group one that is captured only and one that is translocated to understand what's the impact of translocation versus the capture. And we analyze endocrinology. So we have corticosterone, which is for chronic stress, and progesterone to understand the cycles of the females. And recently we started to put these LoRaWAN ear tags that are very small. They're attached to the ear and they're solar. So we also have movement data. And the idea is if we're using these very expensive methods, that is endocrinology, but then we also have the movement patterns through the GPS, in the future, we can kind of use only the GPS data as a measure of stress if we combine the two data. And this is my other co-PI, Cedric Kayale, who's also doing his PhD. He's an ecologist, so we're working together on the same study, on the same samples with 2 different points of views, veterinary and ecology, to try to understand more and integrate more information. This is Shali, she's also a co-PI, she's also a vet. And she also has another affiliated Nat Geo grant. The whole project is supported by Nat Geo. And she is studying some diseases, the Clostridial disease. And the reason is that predisposing factors for Clostridial disease that can bring mass mortality in the animals are basically physiological stress, change in diet, and increased stocking densities, which is everything happens on a translocation. So the plan here is to compare data and understand if the stress of a translocation can produce these, and if can, how can we prevent it? And we also had a veterinary student that had a Morris Animal Foundation on parasites. Also, same here, comparing the chronic stress with parasite load and having a bigger picture. Then we use behavioral score to understand the behavior. Pre-darting is the reaction of the rhino to all the helicopter chase. And then we have scores for immobilization. That is more of anesthetic score to understand if the drugs we use are actually making the rhino sleep. For the recovery, to see if they recover well, if they're competent to be in the wild or in the transport crate. And then we have these other scores for normal behaviors. So we ask the ranger to rate the normal behavior of the rhinos based on this score. And then we have another score and we ask the ranger when they do the monitoring to give us information on behavioral change. And this is again, so we can compare endocrinological data, so the chronic stress and reproductive hormones with scores. So in the future, the government will be able to use the scores to monitor the rhinos. And for example, we found that higher pre-darting scores, so a bigger flight or fight reaction of the rhino during the helicopter chase, was linked to worse metabolic acidosis, but also to increasing cardiac biomarkers. So can actually tell us a lot. And then we have this data collection sheet for the rangers. So this is the head of the rangers of the last translocation we did in June. And every time they see a rhino, they collect information for us about the rhino and also about the status of the samples they collect for us. We also train them to collect samples, to keep them in the cold chain, and et cetera, et cetera. So this is the second part of the project. A lot of data are taken on paper and then they stay there. Plus, we realized talking with the rangers is complicated for them to write on paper and then going back to the office. So in Kenya, they use a lot this app, Ranger, that I don't know if you're familiar, is being used more and more in different fields, but it comes from management of landscapes. So we are creating a module to monitor these rhinos through the app. And the idea is to have different modules for both veterinarians and rangers, because also for us veterinarians, like when I'm there and I'm a researcher and my goal is to take data, but when the veterinarians are in the field, they don't have time to take data on paper and so on. So with an app, it's much easier to not lose super important data. So we have 3 modules, one before the translocation with more baseline information, one during the translocation with all the information about capture, transport, health assessment, behavior, and then post-translocation, which is what we have developed up to now. So the first 2 we're still in process, and include information about space use, settlement behavior, reproductive data, signs of illness. And then we would like to have a way also to introduce like more research data in order, for example, to facilitate analysis of that's the end goal, adaptive management, right? So we are actually in the discussion about how can we make this data ready for adaptive management, and also who owns the data? Is the government? As a researcher, can I have access to the data? Of course, these are rhinos, so it's extremely sensitive topic. So these are all processes that we have to think about. And this is the last part of the most recent part of this initiative, which is more about advocacy for integrating health in conservation translocations practices, but also policy. So we organized a side event last year at the Convention on Biological Diversity COP16 in Colombia, and had different experts from different disciplines, but also policy experts. And we had also Kenya, the Ministry of Tourism and Wildlife, to discuss how can we overcome the lack of monitoring standards, which creates gaps in reporting, but also in resources that go to the ground for us to improve conservation translocations. And we had participants from 5 continents, so it was very inclusive. And the key outcomes were that there is a need to increase One Health collaboration. So for conservation translocation is fundamental, that links wildlife, livestock, human, and the ecosystem. Also, that there is the need to include more traditional knowledge and the communities in monitoring efforts. For example, what we do in Kenya with the rangers, but sometimes maybe this is for the rhinos, but sometimes maybe you have like smaller animals and you can ask the communities to help you to monitor. And then the need for more long-term monitoring to really inform adaptive management, because to inform adaptive management, you need a lot of data and also long-term to really understand the impact. And then we also discussed the need for global data repositories to share health and translocation data that is super ambitious, but recognized as very important. So we launched a community of practice at COP, and the goal is to share knowledge and potential solution to enhance wildlife health for conservation translocations at intersection of conservation, restoration, and One Health objectives. And it's really not running yet, but I show this because I always hope that people are interested to engage and to give ideas also to make this work. Definitely there is a big need to be more active. So if you're interested, please talk to me. I need ideas and collaborators on this. And then this year we did a workshop in Kenya, again with the government of Kenya, Smithsonian, and the IUCN Conservation Translocation Specialist Group and several other partners. And the idea was to have, again, transdisciplinary people coming there and brainstorming what can we do to improve things at global level. Kenya is a very how can I say, has a lot of expertise in wildlife health and also in conservation translocations that is rare to find in other countries. So we thought, let's bring Kenyans there together and let's talk, how can we tell the world to improve things? But also on the other side, the government asked us to use this opportunity to improve things at local level. So we had these 2 kind of big goals. And since we had people from different backgrounds, from government to veterinarians, ecologists, but also we had technical experts and so on, we thought to start with the training from the IUCN Conservation Translocation Specialist Group on decision making in translocation. So everyone could start from the same background. And Axel Moerenschlager, who I know is a Oxford alumni, who is the chair of the group, did the training together with John Yun. And they went through like a few key concepts like SMART goals, differentiating between fundamental and means objectives, using indicators, trade-off evaluation, risk assessment to inform adaptive management, and the need for engaging stakeholders. And actually, parallelly, we had case studies from Kenya, from Kenyans, from different disciplines around conservation translocation. So again, wildlife health, anesthesia, technology, behavior, ecology, but also policy. And the idea was like, if we put a lot of knowledge out there and we do the training in a parallel way, we can really have interdisciplinary discussion and see how we can improve things and think out of the box. So the last day was full day of discussion. So we had a professional facilitator and we used different methods. You can find here the workshop report if you're interested. But the key outcomes were that participants recognize health and welfare as core to planning for conservation translocation. And this is rare. Like I've been to another IUCN training, and usually population goals are what is like seen as core for translocation planning. So even the leadership of the group was surprised that for Kenyans, health and welfare of the animals is core. Like it's not a means objective, it's not a way to get there, but has to be center. And then we did a SWOT analysis, and we found that there are gaps in interdisciplinary collaboration and in monitoring. Then also we recognize the need for technology to facilitate monitoring and greater coordination, and that there are existing information also for policy reporting, for example, also for the Global Biodiversity Framework, but they're kind of hidden. So greater coordination is needed to support that. And we had a discussion on policy led by the Ministry of Tourism and Wildlife, and they managed to have conservation translocation in the Kenya Wildlife Bill draft that is now out for public consultation, and is the first country in Africa that recognize talk about conservation translocation in their legal system. So there was a big achievement. Other initiatives we've been doing through the starts of the community of practice was a forum event at the IUCN World Conservation Congress, again led by Smithsonian and the IUCN Conservation Translocation Specialist Group, with many other partners. And we had a lot of panelists and moderators talking about 4 contentious topics. These 4 contentious topics are the need for post-release monitoring, and how can we also advance individual health and welfare and not only population goals? How can we make sure that the disease management doesn't slow down urgent translocations? And how do we produce the right release candidates, also sometimes from captive breeding centers? And key elements that came out again is like the need for like having more health and welfare in international SOPs, but also more need for monitoring of both source and recipient populations. And for example, also the utility of advanced reproductive techniques to produce viable candidates for release back in the wild. And this is the last event we had last month in Panama at the subsidiary body for scientific, technical, and technological advice. So we proposed an event on conservation translocation, and then the Secretariat merged us with other institutions. So at the end became a very high level event with many partners and many countries. And I found this extremely interesting because we framed the overall goal of the event on biodiversity restoration. And the Global Action Plan on Biodiversity and Health. And we had 4 countries, we had Kenya, UAE, Panama, and Germany talking about their needs and their what they want to what are the opportunities, what are the needs? And then we had expert panels from, again, different disciplines, from WHO to IUCN to smaller NGOs. And we also had donors, the Belmont Forum, discussing all together, how as expert can we support? How can we make things better? So this was extremely interesting, and I'm happy to talk more if you're interested about this. So, okay, to conclude, this is my observation throughout years working in this on this topic. And of course, translocations are super complex, and we need to acknowledge this. So we really need an interdisciplinary approach, and we need long-term cross-sectoral studies to create knowledge to inform the adaptive management. So we cannot expect to learn how to do things overnight. We really need some real investments. And we also need to standardize indicators to have a long-term monitoring that is sustainable. So sometimes we don't have a research, or we don't have research funds, but if we can find those indicators that, for example, governments can keep on using, then we will have a lot of information to learn how to do conservation translocations in a more responsible way. And then, of course, data management. We need to find a way to share data. Also, another lesson learned is logistics and politics. This is an extremely complex topic from also politics. Permits can take a long time. My permit took 3 years. Stakeholder trust. So again, sensitive data for rhinos, but also for other species. So not everyone wants to collaborate, especially with researchers. And also different stakeholders have different fundamental objectives. For me, it's health and welfare. So for someone else, it's something different. So you have to acknowledge these. And then policy gaps, but also opportunities. So there are more opportunities to, get support from policy or contribute to policy. Yeah, opportunities. So this is the UN decade of restoration. And again, restoration should also include the wildlife part, not only plants. So this is a big opportunity. And also One Health is becoming more and more discussed. Also, translocations are increasing. For example, in Kenya now, there is this new initiative called Kenya Rhino Range Expansion that is combining rhino reintroduction, land connectivity, and restoration. And this is the kind of project I want to work on, and well, I am working on, to connect the dots and know more. And so yes, this brings interdisciplinary collaborations also to understand more beyond the animal health and welfare on the impact on ecosystem, on more biodiversity, on the ecosystem, but also on the people and their wildlife, their livestock. And so I believe that a big study, a large study, can also provide a positive proof of concept for policy for linking restoration with species conservation that sometimes are a bit disconnected. So as scientists, I believe we have to go there and provide positive examples. And then, of course, scaling potential. So right now we are on rhinos. There are more resources on rhinos. There are, big institutions, big collaborations. But the idea is to translate what we learn in rhinos on more species and landscapes. So last slide, next steps. I think advocacy is extremely important. So I went to COP30 a couple of weeks ago. And the reason I went is that there was a lot on reforestation, was one of the big topics. So I thought, let me go there. Maybe there is, space for wildlife. And so yeah, I spent the whole week I was there asking people, how can we put more attention on the wildlife as well? So I think it's important as scientists, we go to these big gatherings and understand how to contribute. And then transdisciplinary capacity, this was another side event. I led a COP last year in Colombia at the Women Pavilion. And this was so interesting because it was all women from different backgrounds. Some were party representatives, some were scientists, and all sharing a passion for biodiversity and advancing things. So this was started great collaboration and thinking out of the box. So I think this is extremely important. And then action. So again, we need more positive cases and data and examples. So yeah, that's it. Thank you so much for inviting again. I hope I talked enough about rhinos. I guess you came for the rhinos. So I hope there was enough rhinos. Okay. Okay.
We can open up to questions. Thank you very much. And there's a microphone for the recording that will part of this round. Actually, I'd just like to kick off, and I'd like to understand how in the relative success story, if I understand, of the black rhino in Kenya, what role has been played by translocation, conservation translocation? Is it quite a minor part of what's going on there, or is it a significant part of Kenya's progress in the black rhino?
It's a significant part. So the increase of population for black rhinos in Kenya has been exponential in the past few years.
Mm-hmm.
And the main governmental body for this, which is the Kenya Wildlife Service, really invested in translocations, but to exchange genetics, because these rhino sanctuaries are small. And they're expanding very fast, because if they're not bothered, they actually reproduce well. They don't have problems in reproduction. So they started this program of continuously moving rhinos around, and also now they're opening new sanctuaries. So yeah, it has been extremely impactful in increasing the population.
Can you just give a sense of numbers? How have black rhino numbers changed in the last 10, 20 years in Kenya, or just roughly?
I think they were around, they went down to 300 and something. I think the lowest point was about 20 years ago. I might be wrong, because my background is still veterinary medicine, and this is all very recent for me. So I might not be super accurate in numbers, but I know they went down to around 300-400 individuals. And just when I started this project, there were 800, and this was 5 years ago.
Mm-hmm.
So they're really increasing very fast, and now they're aiming to 2,000 individuals in the next 10 years, and I'm pretty sure that they can achieve this.
Mm-hmm.
Yeah.
Okay, great. It's good to hear of the success of this. Okay, we can open up there. There's a question back there.
Thank you. My question is, many of these translocation projects, they are hesitating to share the data, especially within the first 5 years or 10 years of the project. So that's more or less a sensitive period, because they are scared that the project would go wrong, and they don't want to share the data because obviously they don't want to be scrutinized publicly. And that means that one of the big part of the investment, which is to monitor and do all this coloring and all this stuff, is not really used within the first 5 to 10 years of the project. And by the time they start to use the data, apart from creating just a JPG map, which is based on the daily receiving of the location, the project might have failed. So what do you think we can do in terms of filling this gap for these 5 to 10 years, which is happening a lot? And it's not only in Africa, even in the UK, many projects don't share the data, and they are hesitant to share the data. So how do you feel we should address this gap?
Yeah, that's a very good question, and you're totally right. I mean, this is also what I went through. There was a lot of resistance to, to have a researcher on board and getting samples and data and so on. So I personally believe advocacy, because sometimes if you focus on the national, people feel accused or whatever, but that's why my more recent interest in advocacy and policy a little bit, because I feel monitoring should be mandatory in a translocation budget. It should not be like a researcher interest. There should be someone saying, okay, you want to move endangered animals, in your budget you have to have 10% of budget for monitoring, and you have to share data. But then who is this body saying that? There's no one that can do that. There is the IUCN Conservation Translocation Specialist Group that is doing an amazing job, but they don't have, they cannot impose to countries, or that is mandatory. So I think maybe it's just creating awareness and talking about this, and at some point maybe changing a little bit this culture of fear around translocations.
Thank you.
Thank you very much for this talk. I have 2 questions. The first one is quite detailed. It's about like putting these animals to sleep, but what did you call it?
Chemical immobilization.
Immobilizing them.
Yeah, exactly.
You said sometimes you know in advance which individual you're going to immobilize. Sometimes you don't. Isn't the amount of chemical you need to immobilize, let's say, a bigger animal much bigger than the amount you need for a smaller animal? And how can you so quickly adapt, like the amount you give to the animal that is picked if you don't know in advance which individual that's going to be?
No, that's a good question. So most of the times there is a candidate list. It's just sometimes we can find the animal, you know, like the animal is in a place that is too dangerous to be mobilized there. Maybe, you know, it's close to a river, they might drown, or it's like up a hill or so on. So we go down the candidate list, and usually the candidate list is very wide. Not like we have a lot of candidates. But regarding the body weight, which I guess what you probably refer to in terms of the dose, they're all like in the same range of age, the one we aim to. And sometimes they're not, because sometimes we want to move juveniles, sometimes more like older animals. So what the veterinarian does is having some ready darts with different doses. So I'm not in the team in the helicopter, I'm on the ground team, but my colleagues in the helicopter have like ready darts with, you know, it's like combination and dose for a female, adult male, and juvenile. So they are ready to, yeah.
I see. Thank you. And the other question relates to the individuals that are released. So you're talking about like translocations of individuals that are already part of the population or neighboring populations. How many individuals are translocated from breeding programs that happen, let's say, in zoos abroad? Does that happen?
So not with the Eastern Black Rhino, because we are still in a safe zone in terms of genetic diversity and numbers. So it's mostly like shifting individuals around between because they cannot migrate anymore. We enclose them, so we kind of do artificial migration of the animals. In many species, of course, there is captive breeding and release in the wild, but yeah, luckily I know there are some zoos also from the UK that sent Eastern Black Rhinos, I think, to Tanzania. Okay. But yeah, Kenya has a good population, and for now it's more than enough, and actually very successful to move individuals around and to creating new sanctuaries. So for example, in June this year, there was a reintroduction of rhinos. So there was a ranch, which was a cattle ranch, that decided to convert to conservancy, which is a conservation area. And they took rhinos from 3 different sanctuaries and brought them in order to have, again, genetic diversity. Yeah, so that's the kind of approach.
Great, thank you.
Thanks so much for the amazing talk. I'm a bit curious about what's the plan on the One Health perspective. So you mentioned about One Health perspective for the rhinos. So they're going to be ecosystem health and then linked to the human health as well. So I'm a bit curious is also, because I don't know the concept that much, but I'm a bit curious like pre-location environmental situation and the post-relocation environmental situation. Does that matter to the rhinos' well-being, health status as well? It's like the trees around that, how many grassland it being exposed to, that sort of thing. And then what's the potential reflection on the link to the community human health? Not really, it's like the livestock being eaten or they go like they have some economically suffering, but so something about human health, potentially what's the lens they're going to be look like. Thank you.
Thank you for the question. So wait, let me go back to the first one. Okay, can you repeat the first one? Sorry, I forgot.
Sure. The environmental situation.
So
like when they being like relocated, when they being chased over there, being relocated somewhere else, being released. And then there's the environment
change with the having the rhinos back.
And it's like different environments. Does that matter to it?
Different environment from the source area to the recipient area. So usually there are a lot of assessments before moving the rhinos. Usually the process of getting through all the permits and assessments can take like years. And there is team of experts going in the new area and in the source area, comparing vegetation and other species around and so on. So there is like a very appropriate process to make sure that they will find the right food and the right conditions. But of course, two places cannot be the same. And sometimes these rhinos are moved across the country. So sometimes neighboring conservancies, but sometimes they go a long distance. So I think we need more research also to understand this. It's super important, right? And to understand how different environment play on the rhino adapting, and also the other way around. How is bringing back rhinos influencing the ecosystem? Is this ecosystem changing and so on? And for One Health, so rhinos can share diseases with livestock, but not as much as other species. For example, if you move antelopes, for example, the amount of diseases they can pass to livestock is worrying. So like with those animals, you have to be extreme. And also the other way around, livestock to antelopes. Like last year, bongo antelopes came from Florida in a zoo to Kenya, released. And imagine, like, these antelopes have been like in a zoo for generations, and now you bring them back in the wild, and there are like diseases in goats and sheep and cattle. So there was like a big movement. With rhinos, we are more lucky because they share less diseases, although that can happen. And how it impacts the community economically through their livestock, of course, like if they have morbidity, if the livestock is not producing, or if they die, plus livestock for pastoralist communities is more than money, is more than the income. They're extremely important for their culture. So you really, like, actually my colleagues and myself, but they do like a very they have several teams going out, talking with the communities, making sure the communities are happy to receive the animals, and there's a whole plan. So yeah.
Okay, thank you for the talk. Very cool. I was wondering what have you found out with the protocols now that you are trying to improve in there for the translocations? Have you come to a conclusion really about the safe actions to take nowadays in comparison to how it was done before? And if this has happened already, like, have you seen any change? And the second question is about, you mentioned something about working with a colleague that is working more into the ecology. And I was wondering if you can just elaborate on what is he doing as well that overlaps with the work that you are doing here.
Okay. Yes. So protocol-wise, so let's start that we need we need much more data to understand. So, and translocation don't happen every day. It's like maybe once every 2 years. So what we're doing now, we are doing pilots, but we need more data to understand how to improve things. Things we have improving right now, for example, is the monitoring methods, which is from the anesthesia, how to monitor. Like, this is all new. Like, it was not there. The test we do, the monitoring for anesthesia, like the ongoing monitoring, the pulse oximeter, the capnometer, and so on. So these also help us to have like safety thresholds in the field. We can see if a rhino is in like distress, like physiologically, and for example, we wake them up. We don't move them. Like, we see, okay, this is a value I don't want to see. This rhino might not make it, or might be seriously compromised. Let's wake up. And then also from an anesthetic point of view, we have seen that some drugs that are heavily used in white rhinos are not as effective in black rhinos. So we shifted to another drug, for example, that I've seen with my team with research that instead increase ventilation and increase the oxygen level, which in turn increases many things. Translocation management, something I really want to look into is, for example, the role of bomas. You know, it's like, is it better to just release these animals straight after a transport, or like having them in a boma helps them to adapt and then they can do better? But we need time to do this. It's like we really need time and data, so we're not there yet. And also all the samples we are analyzing, they're still in process, because the translocation was the first translocation we sampled was 6 months ago. Because there was a long stop on translocation in Kenya. There was a big break for internal reasons. And I'm very keen to see, but also the rhinos we sample now will not be enough to really guide this. So that's the conclusion I'm arriving. We need to find ways to make this more sustainable. So we don't always need like big research funds and so on, but we need to simplify what we collect so we can collect big data and really understand. And my colleague, who is an ecologist, so he works for the government. He's the main rhino scientist for Kenya. And so, for example, if I analyze corticosterone and progesterone, he's also interested in thyroid hormones that are more measures for like nutritional stress. And then he's analyzing like vegetation indexes. But most of the analysis we carry on together. And my goal is, because he works for the government, he has been with the government for so many years and he will be there forever. Whereas I'm a researcher, so I'm attached to funds and projects and so on. So my goal is to create for him and the whole institution basically easier health indicators that then he will be able to incorporate in his like ecological monitoring way going on. Because right now what I do is not is very niche, you know, and it's very difficult to understand the health if you didn't study health for many years. So the idea is like, what can I create for the ecological department of the government as data that like in 10 years, when they will be mobilizing rhinos, they can easily say, this rhino had a perfect anesthesia and is totally healthy and didn't get stressed during transport and boma, or like differentiate this rhino was heavily compromised and had some underlying issues, but in a more simple way. So they will be able to continue adaptive management and improving things without having to have super expensive blood testing and so on. So that's the idea. But it's also in process. Like this is like the first study that really is like so much focused on health and long-term consequences of translocation. So even the methods we're in, you know, they're just sitting down and say, okay, let's try these. And yeah, it's a test. It's a test and a lot of lesson learned actually. Any more questions?
Thank you for your presentation. I'm curious about the use of the saliva for the stress when you capture the animals. So I wonder if you have a reference, so how you interpret that sample, whether the animal is more or less stressed, how you can use that as an indicator.
Okay, very good question. It's not validated yet. So it makes sense in theory, so that's why we started collecting it. But so we have a PhD student in endocrinology, and that will be his contribution to the study. Yeah, in theory is that, you know, like hormones have different excretion times in different body systems, and in saliva is around 20 minutes. So if like, it usually takes a rhino like 5 to 10 minutes to go down. So when we are on the animal and we collect the first saliva sample is maybe 10 minutes after the start of the stress. So that should be the baseline. And then if you take another sample after 15 minutes, you should already see a spike. But in most wildlife species, we don't have references and we need to create our own. So.
And then you're measuring on the feces as well.
Yes.
But that
blood
takes a bit, yeah,
a
longer time.
Yes, exactly. So we'll see. We hope to have good results soon, and hopefully next time I can share the results. Thank you.
Follow-up question on that. Can you use, is there any research being done on zoo animals that can be helpful? Like, for example, comparisons, because they are more used to being close to humans and maybe getting saliva samples without needing anesthesia?
Not in the black rhino. So yeah, actually we collected also some samples from a black rhino in Kenya who is in captivity. And so that will be the start of the validation, but no, we need to validate. Yeah. We also, like initially, we also wanted to look into chromogranin A and alpha amylase, which are other indicators for stress, for acute stress. But I think for now we're just leaving them because the validation process was more complicated and so on. So for now we're focusing on cortisol.
Yeah, good.
Thanks.
I think we'll wrap up there. We have a drinks and informal conversations just down the corridor in the next few minutes. So do come and join us if you want a more informal conversation or just have a drink with your friends. Okay, so thank you very much, Francesca. It was really fascinating to see. That's very different from the talks we've had in the rest of the series. So it was really interesting to have that. And, who doesn't love rhinos as well?